Welcome. This is the 86th annual Cold Spring Harbor Laboratory Symposium. This year's theme was biological timekeeping. My name is Rebecca Leshin. I'm Executive Director of the Banbury Center here at Cold Spring Harbor Laboratory. And I'm very excited today to be speaking with Professor Amita Sehgal, who's the John Herr Musser Professor of Neuroscience at the University of Pennsylvania. And she's also been a Howard Hughes Medical Institute investigator for quite a long time, since 1997, I believe. Welcome, Amita. Thank you for joining us.
Thank you for having me.
So I want to kick it off, chronologically, talking about how you got to where you are today. I always think it's very interesting. Everyone has a unique path. And so you can start at birth, or you can start with your science, when you started becoming interested in science, but I'd love to hear about your story.
So quite honestly, I didn't really get interested in science until I was already in graduate school.
Wow, that's an admission.
I did not grow up, with this passion for science as a lot of my scientific colleagues did. I actually liked literature. I grew up mostly in India, where it was all very rote learning education, and so science education was boring. It was very textbook driven.
Mm-hmm.
I ended up sort of majoring in science just because you right thing to do, but not because I particularly liked it. And I ended up in grad school, quite honestly, because I couldn't think of anything else to do.
That's amazing. I love that.
So
but you fell in love with science there.
Yes, I did. And I think what did it for me was being allowed to really delve deep into it, the science, as opposed to having a superficial exposure to it, which is what I had. Like I had very limited research experience, but even what I had was not letting me really get into it at the level that I think one needs to really become passionate about it. But yes, in graduate school, I really did love it a lot.
And I think that's interesting because coming through, it is early on, I mean, in the US as well, where it's a lot of memorizing and learning subjects, and you don't really understand what a scientist is until you get to grad school. So
I think some of that is true here. There is more of an effort in the US to have more like hands-on experiences and to try to relate it to the real world a bit, which did not happen for us. But
So I interrupted you. So now you've discovered your love of science in grad school. And
then how I got into this field was in graduate school. I worked on a human gene, and I realized the limitations of like the human system. And so I decided that for my postdoc, I wanted to go to a genetic model. And Drosophila in those days was, really flying high. So I thought I would do Drosophila, but in keeping with what I did in grad school, I wanted to do neurodevelopment in the fly, genetics of, neuronal development. But it so happens that the lab I ended up in, which was Mike Young's lab at Rockefeller, worked on two projects. So they had a neurodevelopment project, which is what I thought I would work on, but then they had one on circadian timing. And I knew nothing about circadian timing at the time. But when I interviewed there, I learned a little bit about it, and I thought it was fascinating. I'd always, in grad school, I developed a real interest in molecular basis of behavior, but I trained as a molecular biologist at a time when everything was qualitative, not quantitative. You either had a band on a gel or you didn't. You didn't have to quantify things all the time. The behavioral stuff made me really uncomfortable because it was the differences between wild type and mutant were so small, and you need a lot of, stats to prove a difference. And so I ended up, thinking, okay, I'll do neurodevelopment, not this. But the circadian stuff was amazing because the behavior is so robust, and the genetics is amazing, and the mutants have strong effects, and so that's how I ended up with circadian rhythms.
And I think as with a lot of people, I think the behavior is some of that is very easily easy to relate to yourself or to develop an interest in. So it is kind of a little bit like a siren song drawing you in.
Right.
Now I don't, and maybe I'm incorrect in this, I don't necessarily always tie sleep with circadian rhythms, but you do both. Is that sort of the same way there was the neurodevelopment and the circadian rhythms, you have two components, two brains to your lab?
You don't know how happy it makes me to hear you say that you don't always link sleep and circadian rhythm.
Oh, yes.
Because most people don't get that. Yeah, they think of the two processes, sort of synonymously almost. And in fact, they're not. So sleep is a circadian rhythm. It takes place with a 24-hour rhythm, and it takes place at a specific time of day. But there are aspects to sleep that are not driven by the clock. In fact, sleep as a behavior exists because it serves an important purpose, right? We spend a third of our life sleeping, and we would sleep.
Hopefully.
Yeah, and then we're stone. But even if we had no circadian clocks, we would sleep because sleep is important in and of itself. And so on the sleep side of the lab, we're not studying the timing of sleep, which of course is a circadian process. What we're studying is, why do we sleep and what happens during sleep? What makes us sleepy when we've been awake for a long period of time? And so we're looking at the amount of sleep, the depth of sleep, what we are measuring is different, and the questions we're interested in are different. But yeah, most people, when the Nobel Prize got given out for circadian rhythms in 2017, a lot of people called it the prize for sleep. And I'm like,
really?
Yeah.
Wow. Okay. So, well, I'll give myself a pat on the back for already not tying that together. But you said something very interesting, and I also, it was very stood out very much as the first line in your abstract for the symposium this year, which was, I think, function of sleep remains a mystery. And I think, in my background, always thinking about sleep, it was for either memory consolidation or learning. But the work that you do, I think, is sort of more about the physiological functions or the potential physiological functions of sleep. Do I have that correct?
Yeah, physiological, or I guess I would call it more like the cellular molecular underpinnings. So I think we do study memory, right? But we're interested in what it is about sleep that helps, with the consolidation of memory. So people will say, consolidation of memory is a hypothesis. I think it's more than a hypothesis at this point. Sleep does seem to be important for memory in many organisms that have been studied for this. But the question still remains, what is it about sleep that helps memory?
And why do you think it's such a base core function? I mean, as you've already said, you'll do it regardless of a rhythm driving you to do it.
Right.
And yet we don't know its purpose. We know the purpose of feeding. We know the purpose of, other core systems in our body. What is it about sleep that's so mysterious?
Yeah. And I think that in large part, it remains a mystery because until, a couple of decades ago, which is relatively recent in the history of science, people weren't studying sleep to really get at mechanisms. It was a lot of sleep was studied sort of behaviorally, neurochemically, what neurotransmitters are involved. And so it had never been something that cell and molecular biologists or even hardcore neuroscientists were studying, It was a lot of psychologists studied it, right? So sleep a lot of times would get equated with dreams. I mean, yes. I tell everybody this, that I'm in a neuroscience department, and the year 2000, if I had suggested to my department that we recruit somebody working on sleep, they would have laughed at me. I kid you not.
Really?
This was not something that hard scientists study,
Wow. Oh my goodness.
Extreme.
Well, I think you're an outlier in that sense, and then you've added onto it. I think most people, if somebody said you study sleep, they would say, "Oh, she's studying humans, or maybe even cats, or an animal like that." And you're working in Drosophila, and so you've created this model, this Drosophila model of sleeping. Can you tell me about why you work in that model system and how you went about creating a model in Drosophila for sleep?
Right. So I started my work on Drosophila as a postdoc, and that was, from a circadian perspective. I should say too that when I went into the circadian field, and I said I didn't know anything about circadian rhythms at the time until I learned about it in this lab I was considering, but neither did most other people.
It was a big black box.
Oh, yeah. I mean, not only, yeah, with sleep, at least people are aware of it, whether or not, they think it's worthy of research. But with circadian rhythms, nobody knew that this, field exists.
I think I was looking back at the timeline, and I think there's something that starts in maybe the 30s or the 40s, and then there's a jump to the 70s, and then maybe every decade or so there's, and I don't know if it's now hitting the slope in the spike, but you're right. It's not many people know about it, and it seems like it's been slow moving.
Yeah, so circadian rhythms, lagged behind other disciplines, right? And sleep lagged behind circadian rhythms. I mean, both kind of slower to become mainstream. One of my colleagues in the circadian field likes to, tell the story of how one of his grant reviewers, it must have been in the late 80s or something, called it the field of spoon bending.
Oh, is that like Uri Geller, or?
Is that who?
Oh, he was like a magician or a illusionist.
Yeah,
trying to, with your mind, get something.
Right. Right. So circadian rhythms was, so when I was a postdoc, I did a genetic screen. You talked about, jumping to the 70s, which was Ron Konopka's isolation of sperm mutants. And then the second circadian mutant in animals was timeless, which
was you.
Jeff Price and I discovered as postdocs in the Young Lab. And so, we did a genetic screen. But yeah, people, not that many labs worked on rhythms at the time. So anyway, but we studied circadian rhythms, and we realized from the work that we did that Drosophila was an amazing model, and it was the model that led to the elucidation of the molecular clock mechanism. And so when I came here to a faculty position and continued my work on circadian rhythms, I interacted a lot with sleep researchers here, because Penn has quite an enrichment of those. And we started, and they would, say, oh, you guys have done such great work with circadian biology in flies. And can you, in particular, actually, the person who was head of the sleep center here at the time, Alan Pack, would say this. He's like, do you think you could study sleep in the fly? And so I knew nothing about sleep at the time. If somebody had asked me fly sleep, I'd be like, sure, why not? But as I learned more about sleep, I learned how it was, it's different from rest, and it's characterized by specific criteria. And so we started asking, when flies are resting, we use rest activity as a way of monitoring circadian rhythms. So we asked, is this rest in fly a sleep-like state? Is it, for one thing, we knew it was circadian controlled. So that's one of the criteria for sleep. We knew it was reversible. We found out that when flies are resting, they're more resistant to sensory stimulation. So there's what's called an increased arousal threshold. It's hard to wake them, to get them to respond, yes. Right? And then finally, if you deprive them of that state, they have to make up for it. Oh. So it's homeostatically controlled. So that's how we showed that fly rest is sleep. And when I say we, who I'm really talking about is Joan Hendricks, who was a sleep researcher at Penn, who worked on sleep apnea in bulldogs. And then she came to my lab to do a sabbatical. She was a professor here. And she came to learn molecular biology. But while in my lab, she developed the fly model for sleep.
Oh, wow. It seems like it sounds like throughout your career, it's been almost like the principles of improv. It's sort of a just jumping in, yes, and saying, you know, there's not much known there. This is going to be challenging and tough, but let's do it.
It feels like that. Yeah, definitely. I think, and it wasn't necessarily by design. It wasn't like I, set out in science thinking I'm going to work on things that, not too many other people work on, that, we know nothing about. It was just, I would get introduced to them and I would jump into it without thinking. I mean, one of the things I always tell my trainees is that I feel like a lot of times I've jumped into things without thinking too much about them, right? And whether it's projects or whether it's specific experiments. And I said, and in general, that has served me well. And so my lesson, to my trainees a lot of times is, if you think long and hard about anything you are going to do, let's say an experiment, you can always find a reason why it won't work. Sometimes you just have to jump in and do it.
I love that. Oh, that's great advice, especially for trainees, and I'm sure that serves them throughout. But I want to go back to something that you mentioned, which is that there's a difference between rest and sleep.
Right.
And I had written down in my notes, I think with the COVID pandemic, the term fatigue has been thrown around so much as a symptom both of the infection, but also of the vaccination and the immune system response. So I would expect there's a difference between fatigue and sleep as well.
Yeah, so it's really interesting you should ask that question, because we are trying to develop a program in fatigue right now.
Oh, I swear I didn't have any advanced information.
And we had a meeting just this morning about it. And so, because the NIH is really putting a lot of emphasis on fatigue now, they want to get at the basis of chronic fatigue syndrome, for instance. Yes.
Yeah.
So fatigue is characterized by, many different features, sleep being one of them, right? Or sleepiness. But there's also immune disruption or immune molecules changing. Okay. And so our approach to fatigue is actually to think about sickness-induced sleep, which we've actually studied in the fly, and others have and are doing it in mice and worms. And we're finding that, immune molecules are involved in that. And so sleep during sickness is restorative. It helps survival. And so we're kind of approaching fatigue that way, and, link between sleep and immune systems. But yes, so fatigue also tends to involve oftentimes changes in muscle activity. Yeah.
Yeah, no,
it's very complicated.
Yes. Well, I've always said I know the immune system is complicated on its own. So add in this, this other unknown piece, which is sleep, and forget it.
Well, fatigue's been hard to study, which is kind of why the NIH is emphasizing it.
Yeah, absolutely. And I think for some of the people who are describing symptoms of long COVID, it's probably going to be an explosion of need. Right. So we only have a few minutes left. I have two spaces I want to hit before we go. And one is to ask you about the symposium. Were you able to attend any other talks? I know we're in a virtual format, so it was a little bit different, but was there anything, and if you weren't, if you didn't find something new or exciting in any of the other talks, then how did you feel about the virtual format this year?
So I thought I attended every single.
That's great.
And I felt like I learned a lot. I really enjoyed it. I would have preferred to have been there in person.
Yeah. Of course. Yeah.
But, the virtual format worked as well as it can.
Yeah. And I think it's interesting because thinking about sleep too, you think about people's different, some people are better at staying up late or getting up early, and virtual and remote work has kind of made that a little bit more possible for some people. So
yes, that's sure.
So my last question may be controversial. So if you want to abstain from answering, I will completely understand. But as a circadian scientist, I have to ask you your opinion on daylight savings time.
Oh.
Again, if you prefer to abstain, I will understand.
Yeah, so I'm sure I know what most of my circadian colleagues are saying to you, and I see that from a pure clock perspective, we should not have daylight savings time. But
Mhm.
I think that there are a lot of other factors to consider,
Mm,
and so I am reluctant, I guess, to be pushing policy.
mhm.
Yes. Yeah, and a lot of my colleagues are not going to be happy with me for saying it. On the other hand,
Well,
I've said it to them also. 'Cause I feel like there have been other cases where the circadian or the sleep field has pushed for something to be changed in terms of policy,
Mhm.
and then it turns out to then get reversed because
Mm,
so I think there are a lot of things to consider and not.
sure, yeah.
Yeah.
It's not ready to change it yet because we don't have enough information or haven't considered all sides. But I will say it's been split. So
Oh, okay, that's good to know. I thought the whole field was on one side and I was on the other.
no, you've got some allies out there. Well, thank you so much for joining us today. Thank you for taking part in the symposium and then taking this extra time to talk to me. I've learned a lot, and I'm now going to start using some of your advice of not overthinking it and jumping into things. So, so thank you very much, and we'll hope to see you next time in person at the lab.
I hope so too. Thank you, Rebecca. This has been fun.